In this work we provided a proof of very important concept that we can actually target the production of NAD in the bacterial cells, and by doing this kill the bacterial infection. NAD is a very important small molecule in the cell that participates in hundreds of enzymatic reactions, mostly reduction and oxidation. So cutting the supply of NAD for bacterial pathogen would eventually kill it. The significance of this work is actually dictated by the urgent need of developing new broad-spectrum antibiotics. Not many people may realize that we're running short of silver bullets, if you want. There are multiple new pathogens, clinical pathogens, that developed resistant over years against almost all common antibiotics. So there is a huge need to develop new agents that will kill diverse currently known pathogens, emerging pathogens, and including the pathogen that can be potentially used as agent of biological warfare. First, we wanted to find a new target that was not previously explored. And we also wanted a target that would be presented in a broad range of organisms, so that existing antibiotic resistance would not affect the outcome of this program. That's how we arrived to this particular enzyme target, which is conserved and indispensable in a very broad range of organisms, because it's a part of central machinery. It's involved in production of the key molecule that is needed by all bacterial pathogens. This work went for a number of years. We used comparative analysis of multiple bacterial genomes first to identify the best possible target, and that's how we come up with the concept that targeting this particular enzyme, which we call NADD for shortness, is the good target for developing of new potential antibiotics. And then the work included structural analysis, detailed characterization, in silico screening, meaning screening in a computer of millions of possible inhibitors. Ultimately, we came to a smaller set of molecules, and then we experimentally tested them against our target enzymes from various bacterial pathogens, and came up with a handful of chemotypes that look promising. These compounds inhibit bacterial enzyme, they suppress bacterial growth, but they do not affect functionally equivalent human enzyme, which is very important not to have any side effects of these prospect antibiotics. So this is, again, as a key milestone. We need to continue this work. The work is supported by the grant from NIH, from National Institute of Allergic and Infectious Disease. I want to emphasize that this work could happen the way it did only as a result of multidisciplinary collaboration. So not only the wonderful colleagues in my lab, but also our collaborators at UT Southwestern, crystallographers, and in silico designers, or people working in chem informatics in the University of Maryland, contributed to this work.